Yes, now so everyone joined. So let's start this session. Today we will learn something interesting related to the previous one. As previous we have studied about the intra and intergeneric interactions, right? And today we will learn about the chromosomal theory of inheritance, which is little bit interesting as compared to the previous one. So let's try to understand about it. So yes, now the first part of this session started with a very simple questions, and that is identify the structure that inherit the information from one generation to the next generations. Now here you can see cell. The cell is the basic unit of life. Inside the cell there is a lot of organelles, and one of them is nucleus. Inside the nucleus, right? Inside the cell there is a nucleus, and inside the nucleus there is a chromosome, right? Chromosome having a thread-like structure on the surface, that is called DNA, and the particular segment of DNA is a gene. That means, so we can say gene is a particular segment of DNA which is located on the chromosome in the nucleus inside the cell. Yes, are you agree with my point? Just responded. How many of you are agree with my point? Yes. So if this is, then tell me which one is responsible for transfer of genetic information from one generation to the next generation, whether it is cell, nucleus, chromosome, DNA or gene. Which one is responsible? Come on, tell me fast. Okay, and responded chromosome. Why chromosome? Why not DNA and why not gene?
Sir, I studied on the basis of like gene interact with each other, but that gene is present on the chromosome,
Chromosome, yes.
and from one generation to another generation, it's a chromosome that decides everything. So even though gene is the main thing, but the chromosome has various kinds of genes. So in the end, it's the chromosome that decides.
Absolutely. So that means that's why the today's topic is chromosomal theory of inheritance. Means we're going to learn that how chromosome decide, that transfer the genetic information from one generation to the next generation. So yes, it's a chromosome. So please everyone, just keep in mind that chromosome is that structure that inherited information from one generation to the next generation, because this type of concept we have already studied in our lower standard. It started from the 8th 9th standard, right? That the chromosome is the inherent characters, but how that we are going to prove in this particular session, that how different scientists walk behind these chromosomes and they got success to prove that the chromosome is the genetic information that transfer from one generation to the next generations. Let's try to understand in details, but before that, recall, yes, so this is what Mendelian genetics that we have already studied in the last session. We studied about the incomplete dominance, codominance and multiple alleles. Yes or no? Yes, this we have already studied in the last session. If we studied, then who is going to tell me about the incomplete dominance? I'm just going to ask you from all of you, and one of you may have to explain that for me, that what is incomplete dominance, what is codominance and what is multiple alleles. So who is going to explain this one by one? Yes, no one? Okay, so only one student replied. Okay, Anmol Deep said okay. Okay, so and you are comfortable in which concept? Incomplete, codominance or multiple alleles?
Whatever you
Tell me.
say, sir, I think I'm fine with all three.
All three. Okay, but I will give you only one choice.
Yes, sir. Whatever is fine.
Okay, so you can just explain about the incomplete dominance.
Uh, Incomplete dominance is like an exception of the law of dominance. Law of dominance says that the dominant gene is the only one that is phenotypically expressed. But in
Mm-hmm.
incomplete dominance, even the recessive gene and dominant gene are like mixed together. You cannot see complete dominance there.
Right.
Uh,
Yes, yes, absolutely.
it is not the same thing as co-dominance because in co-dominant gene. But
Right.
in incomplete dominance, the dominant and the recessive ones combine together and express phenotypically. In
Okay.
this case, the phenotype and phenotypic ratio and genotypic ratio is the same.
Okay, absolutely right. Okay, then the next, Anmol. Yes, Anmol, you can talk about the codominance.
So, like in incomplete dominant, to like recessive and the dominant, both the character intermediate show their intermediate. But in codominance, like there is one recessive and another one is dominant, but there is nothing like that. They both will show the character in the progeny. Like if we have pure red bull and pure white cow, and by breeding them we doesn't go for F1 generation like or simple red or white.
Hmm.
We get both like roan.
Okay,
We got roan.
that means of you want to say that it is a kind of a phenomenon in which two alleles are both expressed in the heterozygous individual,
Yes, sir.
right? Because they are both express their characters separately in that particular individual. That is what called codominance, right?
Yes, sir.
Okay, absolutely right. Okay, so next number is multiple alleles. Who is going to tell me about the multiple alleles? No one raise a hand in the chat box. Only two raised their hands, so they replied. What about others? Yes, anyone? Anthony, Sasidharan, Sindhuja Sri, can you hear me? Yes, no one. Come on, respond it fast. Okay, well, so Sarjana, are you ready? Okay, please go ahead.
Yes, sir. Sir, from the name itself, we can understand multiple alleles where like two or more than two alleles control a character, sir. Like there will be three alleles governing like same character. So
Okay.
we will be like studying two alleles in a gene, but in multiple alleles, we'll be studying about like three alleles or more than two alleles controlling one particular character.
Okay. Yes. So yes, multiple alleles, codominance, multiple alleles is definitely what you said that related to multiple alleles is little bit I would like to add here that more than two alternate alleles of gene in the population occupying the same locus on the chromosome and it's homologous, they are known as multiple alleles. So you can see here with the simple blood grouping type, like you can see that IA and IB, these are the two groups, and I is a universal which is responsible for the factors. So you can see in basically human beings we have total three type of blood grouping, right? So these are either IA, IB or I, right? So this is the example of IBU blood group related to multiple alleles. So these all three example is important for you that you have to remember. Okay. So this is what about the recall what we studied in the last session. Now let's proceed and try to understand about the today's sessions. Okay. Now as I told you that we are going to learn about the chromosomal theory of inheritance, right? Hello, am I audible to all of you? Yes. Okay, because I think so some it's showing some network reflection. Yes. Am I audible to all of you? Great, great, great. Okay, Kirthini, Sasidharan and Anthony, what happened? Okay, so yes, all of you please focus on this. We are going to start our session that are related to the chromosomal theory of inheritance. But before we have to just focus on that, why we need to study this? Because we have already studied about the Mendel's genetics. Then why we need to focus on this? Because see, in the previous session, you have already learned about the Mendelian genetics, right? But in this session, we are going to learn about the concept related to Mendelian genetics. Means it is similar to that concept that is called the chromosomal theory of inheritance. So you must, you have already studied the structure of chromosomes and cell division from your 11th standard, might be in cell division, cell cycle and cell division, right? So what is interesting in this chromosomal theory? So, initially when Mendel was there, Mendel did wonderful work, but his research is not published because of there is no way, no we can say mode of communications. So in that time communication was not as easy as today. Like if you do any kind of research, you can immediately publish a paper on the internet and everyone know about what you did. But at that time it is very difficult for them. Sir, some glitches it's showing, sir. Okay, what kind of glitches, Anthony? Are you facing any kind of problem? Yes, everyone. Am I audible to everyone right now? Yes? Anthony, please check your internet issues. Okay. So as I said, there was
Sir,
no communication.
sir,
Yes.
were you not audible to him some minutes ago?
Yes, pardon.
Were you not audible to Anthony some minutes ago, sir?
Okay, now but I am audible to you, right?
No, sir, a few minutes ago you weren't audible to me. I thought it was my side of the problem because I too had a technical issue.
Okay.
But I too cannot hear you for a few 2 minutes.
Okay. Okay. So maybe technical issue from your side. As you know, the rainy season is going on, so it may be there's chances that the technical issue may, or either it can be internet fluctuations is there, right? Okay, Bhuvanasri said, sir, your voice is breaking. Okay. Still breaking? See, from my side, everything is very fine. Like we have good internet speed, even a proper audio from my side. But I think so from your side, you can just have your, check your system. Maybe that will be more benefit as compared to, yes, either you can refresh it. That will help you. Yes, and now I'm audible to.
Yes, sir. When you were not audible, I immediately left the session without even my knowledge, sir. It just went
Yes.
out as a glitch. So I rejoined it again. Now you're audible.
Okay, okay. Because I also got a notification like Anne left the session. Then immediately she
Even I
joined the
didn't know
session.
I left the session, sir. It just went out on its own.
Okay, okay, okay. No problem. So hope you will continue. Yes, let's focus on this. We know that this kind of problem is there, but we can have to focus on this. So yes, so what I said that Mendel's did wonderful work, but his work was unrecognized for several years, right? Why it is unrecognized? Because of the several issues, like few are mentioned over here, like communication was not easy as today what we are having that communications. In the previous, in that era when Mendel was there, that time communication was not so easy, right? Second, his mathematical approach was new and unacceptable because, see, Mendel was not a biologist. He was a mathematician. So he used a different mathematical approaches, knowledge, formulas and all. But that time, that mathematician, not going to accept his all these new approaches. So that is one more difficulty from the Mendel's in that era. And the third one, the concept of gene as a stable and discrete could not explain the continuous variations. Means Mendel could not able to explain all these continuous variations, and because of that he could not give a physical proof for the existence of factors. So these are the several reasons that his research unrecognized for several years. So based on his research, right, based on his research, there are three scientists you might have really studied about. The three scientists name is De Vries, Correns and the Tesmark, right? So Tesmark, then Vries and Correns, to all three together, independently rediscover the Mendel's result on the inheritance of characters. So various cytologists have also observed the cell division due to the advantage in the microscope right now, but this lead to the discovery of structure inside the nucleus, right? So in eukaryotic cell, worm cell structure form during the cell division are chromosomes. The chromosome means what? They're a colored body and visualized by the stainings. So an organisms which possess two complete set of chromosome are known as a diploid. That means if they have two set of chromosome, right, XX or XY, so it is a two set of chromosome. It is what it is called, the diploid chromosomes. So a chromosome consists of long continuous coiled piece of DNA in which the gene are arranged in a linear order as we see, right? So the gene are actually, so these gene actually responsible, these are the hereditary units. And chromosomal theory of inheritance state that Mendel's factors have a specific locus, or we can say positions on the chromosome, and they carry information from one generation to the next generations. So this is what the Mendel's idea, which was unproved, and that's why the various scientists work behind this particular theory. And the name first, the first credit goes to this two man, that is Walter Sutton and the Theodore Boveri. Okay, Walter Sutton and Theodore Boveri in the year of 1902. Now, these two scientists, that is Boveri, who support the idea. Boveri was a man who support the idea that chromosome contain genetic determiner. That means of chromosome have something which determine the genetics. So it is a genetic determiners, and he was largely responsible for developing the chromosomal theory of inheritance. Simultaneously, there is a Sutton in the year of 1902, a young American scientist, a student actually, he was a student, independently recognize a parallelism, similarity between the behavior of chromosomes and Mendelian factors during the gamete formations. So they both, when we compile, it's the Sutton's and Boveri independently propose the chromosomal theory of inheritance. So you have to please keep in mind that Sutton and Boveri both independently they propose this chromosomal theory of inheritance. So genes are found at a specific location. They said that genes are found at the specific locations on the chromosome, and that the behavior of chromosome during the meiosis can explain the Mendel's law of inheritance. Clear? So this is about what the initial introductions regarding this chromosomal theory of inheritance. Now, what are the salient features of this theory? What are the salient features, or we can say the important features of this theory? So yes, definitely when we're talking about the salient features of this theory, you can see here. Hello, hello, am I audible? No one responding. Okay, okay, great. So salient features of this theory means what? Chromosomal theory. What is the salient features of chromosomal theory? So very first, when we talk about the salient features, you know, the gametes. Gamete means we have already studied in our previous chapter about the sexual reproduction in human beings, right? Human reproductions. And the gamete means, you know very well. What is gamete? It is a sperm and ova, right? So the gamete, which actually it constitute the bridge between one generation to the next generation. That means of sperm and ova, it form a bridge between from one generation to the next generation. Who is the first generation? Father and mother. Who is the next generation? That is you or the offspring, right? So in between the offspring and their parents, these gametes work as a bridge, right? That means of sperm and ova must carry all the hereditary traits or hereditary informations. First of all, this is the first salient features of chromosomal theory. Clear? So in this image also you can see this is what, this is what, this is a sperm, right? Similarly, this is egg. So this sperm from the male and this is egg from female. Okay, this egg and sperm are actually hereditary information who is going to carry or transfer from one generation to the next generation, right? Now this both the sperm and egg, they are contribute equally in the heredity of the offspring, means both are contributing their equal role in the transfer of hereditary information from their offsprings, right? So that means of a sperm provide only nuclear part of the zygote. As such, hereditary character must be carried out by the nuclear materials. So there is a fusion of sperm and egg nuclei during the fertilizations, right? That means of that nuclear material, that is what during the fertilizations it is responsible. So that nuclear material means what exactly? Nucleus contain the chromosomes, and therefore the chromosome must carry the hereditary traits, or we can say the informations, right? And even this chromosome pair, you know, generally chromosome present in the pair, right? So chromosome pair has a definite role. They have a specific role in the development of an individual. That means of loss of complete or part of any particular chromosome produce structural and functional deficiency in the organisms. That means of suppose any particular chromosome which is responsible for the eye structure, and if that chromosome is lacking or we can say disrupt, then what will happen? That effect is going to be appear on the physical appearance of that particular parent, a particular child, right? So it is what the loss of complete or the part of chromosome which is produced that structurally and functionally deficiency in the chromosome. That means of chromosome is very important, and that chromosome basically it present in the pair, so it has a definitely a specific role in the development of particular individuals. Are you getting my point? Yes. I want response from everyone. Okay, now chromosome, as I told you that both the chromosome or we can say the pair of chromosomes, That means of the gene which is present on the chromosome, they always present in the pair. That means of in a particular chromosome, suppose this is centromere and this is chromosome, right? So it is always present in pair. So present means I'm going to draw one more pair over here. So it is always present in pair. This is a one complete set of chromosomes. Okay. Now what happened? This both chromosome present on the chromosome, you know that gene which is wrapping right on like on this. So it is a gene, right? So gene or the chromosome which is pair, that occurs in a pair in the somatic, or we can say diploid cell. Diploid cell because it is in pair, so we can say it is a diploid cell. So a gamete contains only one chromosome of type, only one of the two alleles of a characters. So we can say that the paired condition of both chromosome as well as gene is restored during the fertilizations. Means initially it is paired, then followed by unpaired, and then it is going to be restored during the fertilizations. So genetic homogeneity and heterogeneity means whether it is XX or XY, dominance or recessive, can be suggested by the chromosomal type and their behavior. That means of if the offspring is a baby boy or baby girl, or if the offspring is heterozygous or homozygous, whether it is dominance or recessive, all these characters are suggested by all these characters. Are you getting my point? All these characters are suggested by the chromosomal type and the behavior. That means of chromosomal type and its behavior. Okay, these two are important that determine the yes, their homogeneity, whether it is homozygous or a heterozygous, whether it is dominance or recessive, all these characters suggested by the chromosomal type and their behavior. Now if there is a homologous chromosome, synaphase during the meiosis and then segregate independently into two different cells, which establish the quantitative basis of segregations and independent assortment of hereditary factors. In many organisms, you know, sex of an individual determine the specific chromosome, and that specific chromosome is called as sex chromosomes. So you might have heard about the sex chromosome, like in man have a XY chromosome, in female XX chromosome. So this chromosome determine the sex characteristics of particular offsprings, right? So it is what? It is nothing but it is a chromosomes. Yes, is this clear? So this is all about the salient features of chromosome. So here what is written? Only the nucleus of the sperm combined with the ovum, and thus hereditary information is continued in the nucleus. Chromatin in the nucleus is associated with the cell division in the form of chromosomes. Another characteristics like
So
any type of, yes,
if genes are present in a specific position of homologous chromosomes,
chromosome, yes.
and after that they interact with each other, the cross happens, allele
Hmm.
changes.
Hmm.
Does it happen then when they are in chromosome state?
Okay, I'm not getting your questions. Repeat your question again.
Will that interaction between genes and alleles
Gene.
happen when it is at the chromosome state?
Yes. See, it's a very simple. Chromosome is always present in the pair, right? If it is present in the pair, that means of gene is also present in the pair, and this gene is responsible for any kind of what we can say deformity. Suppose if it is loss or if it is gain, then you will get an additional advantage, right? Like suppose one simple organisms, any gene or any particular part of chromosomes is missed or it regain, that produce structural and functional deficiency in the organism which appear physically. That will definitely appear, right? Is this clear?
Yes, sir. So can we say that this happens only when a particular nucleus or a cell is dividing? Because that's when the chromosomes form. Before that, it's chromatic, no, sir.
Yes.
So
So
this
it is,
interaction happens only when the nucleus or the cell is dividing.
yes, that division, because cell division is important. Without division, it cannot be happen, right? So division is very important. When the cell division divides, it will definitely happen this chromosomal and all divisions. Clear?
Yes, sir. Clear.
Yes, and
Yes, sir. It is clear.
okay. Now the another one, any type of deletion, as I told you that if any deficiency in the particular chromosome or particular gene, deletion or addition means if there is an extra addition can also be happen, that means of the chromosome cause a structural and functional change in the living beings, means they're going to structurally as well as functionally they're going to be changed. So that means of chromosome is responsible, right? So a number of genes are found in each chromosome, and that gene determine the sex is affected. See, determination of sex is affected by specific chromosomes, and that is called the sex chromosome, right? So this is all about the salient features of the chromosomal theory of inheritance. Now here, chromosomal basis of law of segregation. Now, this is something which is important. Chromosomal basis of law of segregations. Now, in this particular diagram, you can see that what Mendel explained. So, this particular diagram explains the Mendel's law of segregation of Mendelian factor, or we can say the, because Mendelian used the term factor because that time there is no such sophisticated instrument, so he called it as a factor, but now people known as a gene, right? So people known that particular factor is called, now it is a gene. So this particular diagram explains the Mendel's law of segregation of Mendelian factors in term of the movement of chromosome during the meiosis. So you can see how the chromosome move during the meiosis stage. So you can see that this is the this is the particular chromosomes. It undergo a DNA replication. So you know replication generally move in a cell division, because if there is cell division, then only DNA will replicate. You know the S phase, G1 phase, S, you have already know about it, right? S phase, then there is a G1 phase, S, then G2, then followed by there is a M phase, right? So you know that the replication stage, so during that replications, what happened, that it undergo a meiosis, right? So at the time of gamete formations, homologous chromosome bear the factors that is called gene, or the pair segregate in the anaphase 1 of meiosis first. That means of, you can see here, this meiosis first is generally take place in anaphase stage, and you know very well where this anaphase take place, in a M phase. You know M phase is divided into several parts: prophase, metaphase, anaphase and telophase. Yes, you have already studied in your 11th standard. In your lower standard you've already studied this. Yes or no, please respond it. If you know, then I can go from the basic. Yes. How many of you have already studied about the cell division and cell cycle, the phases G1, S, G2 and M phases? Yes, so only 3 students responded. And responded, then Sarjana followed by Bhuvanasri. What about others? I want response from everyone. See, I'm not doing here commentary, right? You are just sitting and just observing and listening. No, you have to respond if I'm asking something. So till now only 4 students responded. What about others? Are they sleeping? See, this genetics is simple chapter. If you try to understand very concentrately, then it will be a very simple, otherwise it will be difficult for you. Okay, now one student joined very late. Jason Devraj, can you hear me? Yes, Jason Devraj, can you hear me?
Sir, I can hear you.
Okay, why you joined late today?
Difficulty in joining, sir.
Pardon?
Sir, I was not able to connect to my Wi-Fi problem, sir. I was having problem with my Wi-Fi bandwidth.
But you joined total 45 minutes late.
Yes, sir.
Why?
Sir, I was, it was, few of them was repairing, sir. Just now it got connected, sir.
See, this is very difficult. See, you are trying for NEET, right? NEET is not such simple that yes, it is just simple. Biology is very difficult. If you not recall every time, no, it is very difficult. It is very volatile subject.
Okay, sir.
So please be joined in the next session. I want you to be joined at exactly sharp 7 o'clock.
Okay, sir.
Here.
Okay, sir.
And even I'm just observing that you are missing last 4 or 5 sessions, right?
Last session I was there, sir.
Are you sure?
Yes, sir.
Okay, then I will check and I will inform you tomorrow. Tomorrow or day after, when there is a session, I will definitely bring your all the attendance report and then I will see you. Okay?
Okay, sir.
Okay, now please focus on this. Yes. So yes, we are on. Yes, G1, G2, yes. Okay. So what I said that at the time of segregation, homologous chromosome bearing the factors, right? That factors pair segregate in the anaphase 1 of meiosis first, producing two daughter cell. So can you see here in the first meiosis phase, it is going to produce the two daughter cell. Yes, this is one and this is two. So this is called the two daughter cell. So they produce two daughter cell. Now in anaphase 2 of, we can say, meiosis 2, the two chromatids of each chromosome, they separate and enter the two new daughter cell. So you can see here it is going to be entered into new daughter cell and they form total how many daughter cells? So you can see total they form a 4, right? So these 4 daughter cell formed have one chromosome along with one allele of gene, or we can say the factor in which is present in it. So thus this is what the daughter cells are haploid and known as a gamete. So you can see here the daughter cell are haploid, here the daughter cell are diploid, but here the daughter cell are haploid, and this haploid is known as a gamete. Okay, this is what the single haploid cell is known as a gametes, right? So this is what the Mendel's law explain the segregation, the chromosomal explanation of the law of segregations, how this are going to be segregated. And you know that Mendel have already explained about the law of segregations, right? So it is based on the chromosomes. Another one, that is chromosomal basis of law of independent assortment. Now how it is different from the previous one? Previous one is a law of segregations, and this is what the law of independent assortment. Now what is in the in this law of independent assortment? So this is also, so this particular diagram or picture we can say explain the Mendel's law of independent assortment of Mendelian factors in the term of movement of chromosome during the meiosis. Okay, so the previous one, it shows the movement of chromosome during the meiosis. This is also indicate the movement of chromosome during the meiosis, but these are, you know, two different pairs of gene or two different pair of chromosomes. How? So let's try to understand here. So in this you can see the two different possibilities which is shown over here in this particular diagrams, right? So yes, you can see allelic gene, or we can consider like suppose there is yellow and green. Here I can mention this is capital A and capital B is written over here, right? This is capital A and here is capital B. So you can just consider this is what this A and B, I can say this is a allelic gene for particular seed color. So let just consider this seed color is yellow and green. So for that can I write yellow and green? So this is yellow and this is for green, B for green. Just imagine I'm not talking about the real concept. Otherwise, if it is yellow and green, then I can say Y and R, yellow and green, right? So this is what the two seed color, just consider yellow and green, are present in one homologous pair. Now the seed shape, round and wrinkled, are present on the another homologous chromosome pair. So round and wrinkled means this is that capital A and capital B is yellow and green, and a small a and small b. This is small a and small b. This is AB and this is small a and a small b. So this is small a and small b. Just consider as a round and wrinkled. Both are present in the homologous chromosome. So total how many? Total one. This is yellow, green, round and wrinkled, right? This is present in one particular homologous chromosomes. Now the parent homologous for yellow and round seed produce gamete which have a chromosome of each type, and therefore they have one gene for yellow color and one gene for the round shape, right? That means of how many characters are there? Yellow, round, green, right, and wrinkled. So total 4 characters are there, right? So this is what the one concept. Another one you can see here in the another segment of this diagram. You can see this is also there, like one of the chromosomes. So we can see here similarly the parent homozygous for green color and wrinkled shaped seeds that produce gamete which have one type of chromosomes, and one having R gene and other having Y gene. So what is happening? These gametes, when F1 heterozygous parents produce 4 type of gamete, and they are individual, you can see produce 4 type of gamete, and that is A, B, A, B, AB, AB, right? So these are the 4 type of gametes, and these 4 type of gametes, yes, I can write here. This is small a, capital B, then there is capital A, small b, and small a, small b. So this is totally different. All 4 are different. So here what it is, it produces 4 type of gametes. So these gamete randomly fuse and give rise to F2 generation with a 4 type of plant in the dihybrid crosses, as we saw in the law of independent assortment, which is 9 raised to 3 raised to 3 raised to 1, right? That is we have observed in Mendelian genetics, right? So this is what the how it is going to be independent. This law explain the law of independent assortment, how they're independently assorted, right? So these two law very nicely explained the chromosome which is responsible and chromosome that explain the law of segregation and law of independent assortment, right? Yes, is this clear? So this is what just a theory proposed by Sutton and Boveri, right? Is this clear? Okay. Now, this theory was explained by Sutton and Bowery, but the experimental verification of this chromosomal theory was actually given by the Thomas Hunt Morgan. Okay, he is another scientist who work on the Drosophila melanogaster. It is a fruit fly. You can see this is a fruit fly. Generally, uh, 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 this kind of bees are called the fruit flies. They generally love fruit, so they generally depends on the fruit to survive. So that's why it is called the fruit fly. And the zoological name of this particular species is called the Drosophila melanogaster. Okay, so it is written over here. It's a Drosophila melanogaster. Okay, so that is the name of the scientist called the Morgan, right? Thomas Hunt Morgan, yes. So this Thomas Hunt Morgan's, actually it is called the father of experimental genetics. Means he is also called as a father of experimental genetics because he proved that chromosomal theory with the experimental proof, and that's why it is called the father of experimental genetics. He selected fruit fly Drosophila melanogaster as an experimental material because this Drosophila melanogaster is very small in size and it have various advantages as compared to the pea plant, because Mendel selected pea plant as a model organisms, but Morgan selected Drosophila, which having a lot of good advantages as compared to the pea plant. So it have extra advantage on pea plant as compared to Drosophila. So Drosophila he selected on studied on Drosophila. Now there are several reasons why this Drosophila is more advantage than pea plant, because Drosophila, kind of fruit fly, and it is easily available uh, in any kind of fruits, like we can say mango, banana fruits, where it feed over yeast cell present over the fruit surface. Means if there is a fruit surface, on the fruit surface there is a lot of yeast cell, so it generally grow on that yeast cell, right? So we can say it is very easy to cultivate, it is very easy to find, it is easily available everywhere. Second things, these flies can be reared inside the bottle. That means of if you want to uh, 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 harvest this particular species, or you want to multiply the if you want to increase the number of a large number of this fruit fly, use a bottle. That means of in a particular milk bottle you might have seen. In that milk bottle you can just take a yeast agar, or you can just put a yeast agar powder inside it. Okay, and you have to open it for few days. Drosophila will start growing in this. They start laying their egg inside and it will start growing. So within a week you will get ample amount of Drosophila from this particular bottle. Means it's very easy to cultivate, right? It is very easy to cultivate. So it is easily cultivated in the bottle inside, because nurture in the bottle over the media containing containing cream of wheat or molasses or any agar, it can be easily cultivated. So second thing, it is easily available. First things first, and second thing, it is easily cultivated in the bottle. Now third important what can we can say advantage of this Drosophila is a new generation can be raised within a 2 weeks with a single mating producing hundreds of individual. Means if there is one male and female Drosophila inside the bottle, that Drosophila one time mating they produce hundreds of egg at one time, right? That's why its cultivation is very easy as compared to the pea plant, because pea plant take at least 3 months, but here it is just cultivated within a week, within a 2 weeks. Are you getting my point? Why this Drosophila is well organisms as compared to the pea plant? Yes, are you getting my point? Okay, So this is three important what we can say advantage. There are several other advantages also we can say, but these are the very important. Breeding Drosophila is quite cheaper as compared to the cultivation of plant, because cultivation of pea plant required lot of labor work than what we can say, all the atmospheric condition is required to grow that pea plant. But Drosophila cultivation is very cheaper, means no costly what we can say media is required. It is easily cultivated in a particular bottle also. That means of it is very easy for as a study material organisms. Now this is the characteristics of Drosophila, and that's why he selected fruit fly Drosophila to prove the chromosomal theory of inheritance. Now, experimental verification of theory. So how actually he proved that the chromosomal theory of inheritance based on this uh, particular organisms. So yes, he could be grow on the simple artificial media in the laboratory, means this is what I said about the advantage that this Drosophila can easily grow. It's a simple artificial media. It can grow. The life cycle is very short, means it can grow with it can be only just 2 weeks. A single mating produce how many large number of flies? That means of single mating it produces 100 flies at a time, and has a 4 pair of chromosomes which differ in their size, means they have also 4 pair of chromosomes and all 4 pairs are different in size. So there was a clear differentiation of sexes, means they have clear male and female sex organs, that male is smaller and female is larger. As you can see here, this is the male and this is female, right? This is female. Female eye is more attractive, you can see the red, and male eye is white, you can see this is a male. The symbol is also indicating. So it has many type of heredity variations which can be easily seen through the low power of microscope, and that's why he select the Drosophila as a model organisms. Now see his finding, how he proved that. So he selected the red eye female which is a wild type, and white eye male which is a cultivated. Now this red eye female and white eye female when cross, it produces red eye female, 2 red eye female, 1 red eye male, and 1 white eye male. That means of the ratio is 2:1:1, right? So the eye color gene was inherited in the different pattern by male and female. So you know the eye color, you can see here it is a white eye, this is a red eye color, this is white eye color. So the male flies have a X and a Y chromosomes, that is XY. So they have XY chromosomes, while in female flies have two XX chromosome, means they have X and X chromosomes. Now X make the difference. X make the difference means you know that XX and XY. When it combined, this can be a XX, this can be XY, this can be XX, and this can be XY, right? So eye color gene on the X chromosome correctly predict that all the female flies will have red eye, while half of the male flies will have a white eye. And this can be proved through the sex linkage. So he proved that that how the chromosomes linked with that particular organisms, that chromosomes basically it inherited the character from one generation to the next generation. Okay, so is this clear about this Morgan experiment, about the chromosomal theory of inheritance? Yes, please respond it fast. Okay. Now sex linkage. Now what is linkage? Now here linkage means what? Let's try to understand. Linkage, the name itself indicates linkage means it is the tendency of gene to stay together, tendency of particular genes. Like suppose there are two genes, so they have to stay together during the inheritance through a generation without any change or separation due to their being present on the same chromosomes. That means of if two genes are present in the same chromosome, they have to be linked together through during the inheritance through the several generation. Means suppose in your generation the chromosome are linked, they have to link through generations, and that is what called linkage. So gene located in the same chromosome are linked gene, and those present in the different chromosome, they are termed as a unlinked gene. Means linking gene means now it is clear, right? Means gene is staying together during the inheritance through the generation without any change or the separation, that is called linkage. And this linkage location on the same chromosomes, either it can be located on the same chromosome, okay, and if it is not present on the same chromosome, it is called unlinked gene, right? So they have to present on the same chromosomes. And this type of linked gene one of linkage was first discovered by the Bateson and Punnett in the year of 1903 in the 6 in the sweet pea, and first proved by the Th Morgan. This is not a feast, it is first proved by the Th Morgan. Okay, so actually it is proved by the Th Morgan. Clear? So actually this Bateson and Punnett, they just discovered on the sweet peas, or we can say Lathyrus odoratus is a kind of sweet pea, and in that they both discovered that linkage. However, it was Morgan in the year of we can say 1910 who clearly proved and defined linkage on the basis of his breeding experiment on the fruit fly, that is what Drosophila melanogaster. So actually for linkage he also used the same organism as he used in the chromosomal theory, that is the Drosophila melanogaster, because chromosomal theory related to the link, right? So how this chromosome theory of linkage, they are linked, that he proved. So he actually state that, hello, yes, anyone want to speak something?
Sir, is the linkage on the same chromosome or the linkage between two different chromosomes?
See, no, linkage means on the same chromosome. If it is on the same chromosome, it is called linked gene. Okay, and if it is present on the different chromosome, then it is called the unlinked gene.
So in like previous slide, both the capital A's of that color chromosomes are linked, sir.
I mean to say, in this slide.
Yes, sir. Is it
Okay.
both capital A's are linked or capital A and small
No,
a are
see,
linked?
this is like we can say this is from the dad and this is from the mom, right? So these are two homologous pair. If these two homologous pair, there is a link, there is gene which is a link, right? So this is what this linkage, they are going to uh, link together throughout the several generation. If they are linked, then it is called the linkage, and if it is not linked, then it is called the unlinked gene. It's simple.
Which is linked with what, sir?
See, linkage means what? Like suppose this is what, let me tell you, this is one particular chromosome, right? Just focus, yes, and please focus over here. This is linkage, and this is one particular character, or we can say the trait or alleles, I can say. This is alleles A and this is alleles B. Okay, and these both are the kind of, just consider these both are the gene. So these genes should have to be together throughout the generation. This is the first generation, This is the second generation. So they have to be linked throughout the generation. If these two genes are linked throughout the generation, it is called linkage. And if it is not linked, means here it is linked, but in the first generation they get segregated. Suppose this is one and this is another one, segregated, and it linked with another one, means it is unlinked gene. Because here it is what? It is linked, and it is continuing in the first generations, it is continuing in the second generation, it is called linkage.
So basically the position of the genes should be the same throughout
Same,
the
yes.
generations, but
Yes,
it is
the
called
position.
linkage.
Yes, positions and even their structure should be same, right?
Yes, sir, clear.
So position, see, always keep in mind, linkage is a kind of tendency, or we can say the capability of gene to stay together, right? It is to stay together during the inheritance through the generation without any change or any kind of separation. That is what called the on the same chromosome, not on the different, on the same chromosome, then it is linkage. And if it is on the different chromosome, then it is called the unlinked gene. On the same chromosome, it is called linked gene. On the different chromosome, it is called unlinked gene. Means suppose there is no tendency, they get separated. On the different chromosome, it is unlinked gene. Clear?
So then what if it was unlinked in the first generation and it was linked again in the second generation?
Yeah.
Then what kind of
Let
gene would it be?
let me explain here for you again. See, linked gene means what? Actually, linked gene generally occurs on the same chromosome. Always keep in mind, it is always present on the same chromosome, and do not shows independent assortment. Means they're not going to independently assort, but they remain together and are inheritant. And because of that, they being present on the same chromosome. And that's why we saw the the dihybrid ratio in a monohybrid crosses, that is 3:1. In a monohybrid crosses, we saw that there is a 3:1 monohybrid, that is 3:1. Why 3:1? Because they have linked together, right? And when we do a test cross, their monohybrid test cross, we got the result like 1:1. That what indicate? That means of gene are linked, right? In case of unlinked gene, unlinked gene means what? They basically present on the different chromosome and shows independent assortment. That means of unlinked gene is always present on the different chromosome, and they have to show independent assortment. And that kind of ratio which we generally found, that is 9:3:3:1, right? And in the both the crosses, 50% parent type, 50% recombinant generally observed in the unlinked gene. So this is two important concept which I think so you all should have to remember. What is linked gene, and what is unlinked gene. Unlinked gene. Okay. So linked gene, what I said, that the gene which present on the same chromosome, that means of genes present on the same chromosome, and they do not show independent assortment. Even it is not showing the independent assortment, right? Assortment. So, and unlinked gene means what? They present on the different chromosome. Here it is the gene which is present on different chromosomes, and it is independently assorted. This is independently assorted. That's why they are called the unlinked gene. Yes, clear? And
Yes, sir, it is clear.
and
Yes, sir, it is clear.
okay, good. So yes, so linked gene means gene on the same chromosome do not show independent assortment, remain together and inherited. Unlinked gene means what? Gene on the different chromosome and so independent assortment. So this is what the linked gene. This is the gene 1, gene 2 which is linked together. This is not linked because only one gene are present and another gene present over here. So this is not a linked gene, right? Now this linked and unlinked gene, or there are various type of linkage, but we have to learn about there are two type of linkage. That is, one call it as a uh, complete linkage and incomplete linkage. Now what is this complete linkage and incomplete linkage? See, complete linkage means what? It is a process when two or more gene on the chromosome are close to each other, and if there are two or more genes, they are very close to each other, they have to inherit it together in the next generation. And if they inherited in the next generation, this process without crossing over between the gene are called complete linkage. Okay, so it is of two types. One is called the complete linkage, another one is called incomplete linkage. What is complete linkage? Complete linkage means what? Two or more gene when present on the chromosome, they are very close to each other and they have to inherit it together in the next generation without any crossing over. So you can see here in this diagram, this is what the black body, purple eye, gray body, red eye, two different type of what we can say flies. Okay, now their gametes you can see. So in the first generation, what you will get, there is a no any kind of crossing over, like they are just cross their gametes, and the offspring which produce gray body red eye, black body purple eye, but there is no any crossing over in between. That's why you can see the all the character gamete which is passing in the F1 generation, it is not going to be changed. As here you can see red and G, here is also red and G, red and G, that means of that indicate it's a red eye and gray body. So red eye gray body, red eye gray body, red eye gray body is also passed in the first generation. That means of here there is no crossing over take place, right? So this is what the complete linkage without any crossing over. But in case of incomplete linkage means what? The gene are not too close to each other first of all, and little crossing over take place between this gene. That means of if there are two genes, there is a crossing over, possibility of crossing over. So you can see here the black body purple eye, gray body red eye. When there is a crossing over, you can see crossing over means what? There is a crossing over in between these two gray body and red eye chromosome. What happened? See, the red eye cross with the G, it become R and G, that is gray body, and the red part of this section joined with another segment, right? So this is what the crossing over. So when the crossing over take place, what happened? The crossing over take place between this gene and they segregate and are inherited independently. That means of once there is a crossing over, they are segregated independently. They are not going to segregate together. They are independently, and that's why it is called independent assortment. And thus a new type of combinations are produced in the successive generations, and that's why it is called the incomplete linkage. So it is a complete linkage, and the another one is called incomplete linkage. Yes. So this is what the two type of linkage, complete and incomplete linkage. So in complete linkage, you will get a 50%, means 1:1 in F1 generations, or we can say 50%, 50%. But in incomplete linkage, you will get, see, 47%, 3%, means it is not completely 1:1%. Clear? So this is what the complete and incomplete linkage. So complete linkage is it never form a new combinations. That means of in the complete linkage you will not get a new what we can say combinations, right? Always you will get the same offspring with their parents. Crossing over is absent in it. That means of crossing over is not found in the complete linkage, and such type of linkage are very rare in the nature. Okay, so this complete linkage is very rare in the nature. Male Drosophila and female silk moth is the best example of this complete dominance. Complete linkage. Complete linkage. But in case of incomplete dominance, new combinations are formed due to the crossing over, because there is a crossing over in that incomplete linkage, and because of that there is a chances of new combination will arise. The percentage of new combination is equal to the percentage of crossing over, which is less than the 50%. So you can see in the in this you will get a only 50, less than 50%, means only 47% we saw. So it is less than 50%. Even 3% it is also less than the 50%. So the crossing over, the percentage of crossing over is less than 50%. And the best example for this incomplete linkage, you can see the eye color and wing size of the Drosophila. So eye color and wings size of the Drosophila is the perfect example of incomplete dominance. Clear? So here you can see how it is calculated, the percentage recombinations. So you can see this is white eye miniature wings. The number is 359, and this is 391. So total combination male and female. So total combination is 750. Similarly, in wild type you will get 439 male, female, and 352 male. So total combination is 751%, right? So total parental type is, when we add 791 and 750, it become 1541, which is a total parental combinations. Means it is matching with their parent. But the offspring or the recombinant, or we can say the new variations, new combinations is what? So wild type wings is 218 female and 218 and 257 in male. So total become 455. That is a new combinations. Another one, that is wild type with miniature wings, which have 335 and 210. So total become 450. So total new combination you can see here, total 900. So when we add all both three, that total progeny which is produced in the F2 generation, when we calculate it become total. So adding this 551 and 900, it become total 2441. So total combination is 2441. So if you want to calculate the percentage of recombinations, so how percentage of recombinations? So we want to find only recombination, not a parental phenotypic character. We want to find out the recombinant. So how you will get calculate? So the percentage of recombination is total recombinant upon total progeny into 100 into 100 because we have to calculate the percentage, that's why we multiply 100. So what is the percentage? So you have to just write the total recombination is how much? 900. From you will get the 900 total recombination. Total progeny, how much we calculated, we added, we got 2400, 2441 multiply by 100. So if you just divide and multiply by 100, you will get the percentage, which is 36.9%. So this is how we can calculate the percentage of recombination. Is this clear? So that means of you have to remember the formula, percentage of recombination is equal to total number of recombinations upon total progeny multiplied by 100. 100 why? Because we are calculating the percentage, so we have to always multiply by 100, so we can get the percentage. So that's why we multiply 100, and this is the formula which you need to remember to calculate the percentage of recombination. Yes, is this clear about the incomplete? So always keep in mind the recombination you will always get in an incomplete dominance, not in a complete dominance. Yes, tell me why. Why I said the percentage of recombination always you will get in a incomplete linkage, not in a complete linkage. Why? Very simple questions and you have to respond on it. Why?
Because the genes are little far from each other, sir. But in complete, they are so close to each other that it's hard to separate them. And if they cannot be separated, recombination cannot take place. Whereas in incomplete, they are a little far away from each other, so easily separated.
And you forget the one important point, that is crossing over,
Yes, sir, crossing over.
right?
Without crossing over, recombination will not take place, sir.
Yes, so crossing over is very important. If there is a crossing over, then only you will get a new combination. And if there is a no crossing over, that means of there is a no particular combination. And that's why this percentage of recombination you will always calculate in a incomplete conditions, not in a complete conditions, or we can say linkage. Clear to everyone? Yes, Jason, Devraj, please respond it. Is this clear?
It is clear, sir.
Okay, any doubt in this? Yes, before I'm moving with the next part, just confirm that everything is clear to everyone till now. Or still if you have any doubt, then please ask me. Yes, Kirthini, what are you doing? Kirthini, what are you doing? Beta, please respond it. What are you doing? Are you chatting with the mobile, with your friends? Show me your hand like this. Sure. But I can see the mobile on your desk. Am I right? Kirthini, please respond it. Yes, Kirthini.
Sir, I'm just having my notes, sir. That's it, sir.
Okay, no mobile with you.
Yes, sir.
Are you sure?
Yes, sir.
Okay, then that's great. Okay, so please respond it in the chat box that everything is clear to everyone. Respond it fast. I'm waiting. Okay. So Anne responded, Kirthini responded. Okay. Well, let's move with the next part, that is factor affecting linkage. So now we studied about the linkage and type of linkage. Now let's move with what are the major factors that are responsible for affecting the linkage. Yes, so there are four important factors, that is one is age, temperature, radiations and the distance. These are also responsible for linkage, right? These are the important, we can say the factor which is responsible for linkage. So let's try to understand in detail that how these particular factors affect. If we're talking about the age, so how age affect the linkage? Actually, no, the advancing age, the chances of crossing over are reduced. Means if any particular organism, their age is more, right? So the advancing of the age of, we can say the chances of crossing over are reduced and hence strengthen the linkage increase, right? So that is one more factors. Temperature, rise in temperature increase the chance of crossing over. If temperature is high, there is a chances of crossing over, right? Increase the number of chance of crossing over, therefore decrease the strength of linkage. So if there is temperature high, chances of crossing over take place, but there is disadvantage that decrease the strength of the linkage. Radiations also affect the linkage. Rays, even the X-rays decrease the linkage. Radiations cause recombinations and crossing over. Means radiation is something important. So radiations, let me write here. Radiation means what? Radiations like UV ray and X-ray. UV rays and X-rays. Okay, these two important rays decrease the linkage. It basically responsible to decrease the linkage and radiations cause recombinations and crossing over. That means of crossing over and linkage, or we can say the new combinations, new recombination take place with the help of radiations. Means radiation is important for the crossing over as well as the new combinations. And another factor that is called distance. So increase the distance between the two gene, decrease the percentage of linkage. If the distance between the two gene is far away, one from the one end, one gene from one end, another gene from the next end, so definitely the percentage of linkage is decreased. So percentage of linkage is always decrease. If two genes are very close to each other, then there is chances of linkage. Percentage of chances of linkage is increase. So that means of distance is also determined. This is also the important factor of linkage. So you have to remember that important factor affecting the linkage, that is age, temperature, radiation and distance. These are the four important factor which is responsible for linkage. Now, what are the significance of linkage? So yes, definitely linkage reduce the chance of formation of new combination of gene in the gametes. Thus, it helps to keep the parental racial and important trait together. Means in a linkage, we can say it reduce the chances of formation of new combinations, right? And it help in maintain the important trait of newly developed variety. Because if there is a new variety developed, so that is going to be maintained. It helps to maintain that new variety trait in the several generations. That is what the important significance of linkage. Even it also allow that it give important informations about the location of gene in the chromosome. That means of where the important genes are present, that information is given by this linkage. So it is also good. Even it also disallow the plant and animal breeders to combine all the valuable trait in a single variety. Means you cannot uh, 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 what we can say, enhance a particular organism's trait which is totally unique. Like suppose uh, any particular organ, suppose I want a body like Salman Khan, right? You know very well who is Salman Khan, right? Yes, Bollywood star Salman Khan, you know very well, yes. And he's famous for his body. Even John Ibrahim is also known as based for his bodybuild, right? So if I want to that particular gene who make the body stronger, that gene you cannot accumulate in a particular trait. Okay, so actually it is disallowed by the linkage. Are you getting my point? So this is what we can see that how the linkage is significance. So as given over here. Now the sex linkage. Now what is this sex linkage? Okay. Now before moving with the sex linkage, this is the next topic. So now I would like to give you break. So just tell me how many of you are interested for the break. Otherwise I can continue or I can give you break for 3 minutes, 3 to 5 minutes at least. So just responded how many of you want a break? No one? And Jason, Kirtini, Sasidaran. Okay. So let me generate the poll. So I just want to know how many of you are interested for the break. So yes, poll. Break. And here I'm writing yes, no. Okay, so I'm generating the poll for you all. Yes, poll is generated. Okay. So only 50% responded. Okay. 57% now. So it is more than 50%. So yes, I'm going to give a break for 5 minutes. So you all can take a break and then we will continue from the sex linkage onwards. Okay. So you all can take a break. Hello. Hello everyone. Am I audible? Yes, break is over now. Hello. Hello everyone. Am I audible? Okay. Well, so let's come to the linkage, but related to sex linkage, how these sex linkage are uh, responsible. Let's try to understand what is this linkage. Now, so far you have studied well about the linkage and what. Now here the sex chromosome, you know that we have in human beings or any kind of living organisms, especially we can say the animals, right? They have sex chromosome called X and Y. So XX present in female and XY present in male, which carry the gene for sex determination. Yes and no, because that XX and XY determine the sex of the offspring, right? So they actually carry the genetic information for the, they are called the carrying gene for the sex determinations. So some gene for some somatic character also. That means of all these chromosome is not for the sex determination. Few of that chromosome are responsible for somatic character. Now what is somatic character and what is gametic character? You know somatic and gametic cell? We have two type of cell in our body. Somatic cell and gametic cell. Yes, somatic cell and gametic cell. What is somatic cell? Yes, anyone? Anyone know about this? Somatic cell and gametic cell?
The cells that are not related with reproduction or anything related to that, not germ cells, everything other from that is somatic
Yes,
cells.
yes. So somatic cell means except the germ cell, or we can say except the sex or cells. We can say the sperm and egg, right? These are the gametic cell. And somatic cell means except the sperm and egg, that is called the somatic cell. So all our body are the somatic cell. They follow a mitosis division, and gametic cell follow the meiosis division, right? So so the gene for the somatic character present on the sex chromosome, they are called sex linked gene, right? The gene for the somatic character present on the sex chromosome are called sex linked gene. And the character which are controlled by the sex linked gene are termed as a sex linked characters. And when their inheritance, that is known as a sex linked inheritance, right? So the location in the sex chromosome, that is called the sex linkage. The location, their location of the sex chromosome is called sex linkage. So you can see sex linkage when the gene of somatic character are present on the sex chromosome is termed as a sex linked, and the location, that phenomenon is referred as a sex linkage, right? That means of where that character present in that sex linked gene, that phenomenon is called the sex linkage, right? So basically the two important sex linked human disease that are called hemophilia and color blindness, which is very common. Hemophilia and color blindness is very common, which is referred as a sex linked human disease, right? So here you can see the one character. So yes, so this type of sex linkage, so there are basically two type of sex linked gene. One is called the X linked and the another one is called the Y linked gene, right? X-linked and Y-linked gene. So X-linked, inheritance of X-linked characters. So for example, we can say the hemophilia or color blindness, which is related to the X-linked gene. And for Y-linked gene, the characters are located on the Y chromosomes. They are the polyandric characters, and they're called the common example for this type of Y-linked disease are hypertrichosis. So hypertrichosis is a kind of Y-linked disease, and hemophilia and color blindness is a X-linked disease, which is generally found in human beings. It related to the X and Y linked characters. Even in case of Drosophila, you can see this is also kind of X-linked gene, which is generally found in the eye color in Drosophila. So eye color in Drosophila is also a kind of sex-linked linkage inheritance. Clear? Now, what are the important characteristics of this sex linkage? So here you can see the first of all, mother passes the alleles of sex-linked trait of both son and daughter. Means mother, if having that disease, mother is responsible for transferring that disease to their son as well as daughter. So majority of the sex-linked traits are recessive. Always keep in mind, sex-linked trait are recessive. Majority is recessive, right? Second, third point, sex-linked traits are more apparent in the male than the female. Means sex-linked disease are more. It's very commonly observed in male as compared to female. So as many sex-linked traits are harmful, males suffer more from the sex-linked disorder. Means especially males suffer if he may suffer from these sex-linked disorders. Females are generally function as a carrier. Means female basically work as a carrier, so they just carry that disease from one generation to the next generation. So they are act as a carrier. They are not suffering from such kind of disease. So because of that, the recessive genes can express themselves in the female only in the homozygous state, and that's why, because you know that female is always homozygous because they have XX chromosome. So in the XX chromosome, this sex-linked disorders is going to be suppressed, and because of that, the mother is female is act as a carrier, but male is act as actually we can say the suffered from that particular disease. Okay, so this is what the characteristics related to the sex-linked diseases, that is called the X-linked disease. Is this clear about the sex linkage? Okay, so this is sex-linked characteristics of sex-linked inheritance. Now the another concept related to the linkage, that is called the linkage map. Now what is this linkage map? So now we already studied about the linkage, right? So linkage map means what? To measuring the distance on something related to. So yes, linkage map is what actually linkage map. Generally, it is a linear graphic representation of a sequence or relative distance of the various genes present in the chromosome. So here you can see in this particular chromosome, this is a what? This is a chromosome, just consider, and here is three different genes. This is A, this is B, CN and VG, is three different gene present on this chromosome. So when we measure the distance between these three, between this B and CN, or between the B and VG, or between the CN and VG, that distance is considered as a linkage map. So what are the actually important steps for the linkage mapping? What are the basic background of this linkage? Actually, you know, gene basically present in the chromosome. Just a minute. Yes. So basically gene present in the chromosome, right? So what happened? The gene present in the chromosome are arranged in the linear sequence as you can see in this diagram. So the frequency of crossing over take place over here, and hence the recombination between the two genes is directly proportional to the physical distance between the two. So in a simple word, if I say that two genes which are nearest to each other in the chromosome will have the least crossing over. That means of if suppose there is a two genes, suppose B gene and CN, which is very near to each other, then that means of there is no crossing over take place, no crossing over take place in this two gene. Why? Because the distance between these two genes are very least. Clear? Are you getting my point? Simply in other hand we can say if two genes which have the maximum distance between them on the chromosome will have the maximum percentage of crossing over, or we can say the recombination. So you can see suppose B and between B and VG. So you can see in this in these two gene the distance between is too far. So there is a maximum percentage of recombination, right? That can be observed. That means of if the two genes are very close to each other, there is a least crossing over. If they have to present, if they present on the two different in a long distance, or a maximum distance between them on the chromosome will have the maximum percentage of crossing over, or we can say the recombinations. And therefore the relative distance between the gene is indicated by the percentage of their recombinations, or we can say the crossing over. And because of that, chromosome is chromosome mapping, or linkage map is also called as a chromosome mappings, right? So here actually this linkage map, it Alfred and Stuart Wendt used the recombination frequency between the gene pair of measuring the distance between the gene and mapped their positions on the chromosome, right? So genetic map are used as a starting point in the sequencing of genome. Generally it is used in the human genome project for genetic mapping, right? So this is the applications where generally this linkage map is used. Okay, now let me tell you some important points on this, that in this, suppose there is a 1% crossing over between the two linked gene, right? If there is 1% of crossing over between two gene, that is called known as one map unit. It is called one map unit, one map unit, or you can call it as a centimorgan. One map unit, or you can call it as a 1 capital M. So C for centi, M for Morgan. Morgan, you know, TH Morgan. Yes, all of you know about the TH Morgan? Yes or no? We have just studied about the linkage, relation between the linkage. Yes or no? Yes. So that's why if there is a 1% of crossing over means 1 centimorgan. And if there is 100% crossing over, if the 100% crossing over, that means of that is termed as a Morgan, complete Morgan. If there is a 100%, then it is called only Morgan. 100% means Morgan, 1% means one map unit or 1 centimorgan, and if there is only 10% crossing over, so that is called the decimorgan. If 10% is there, so I can call it as a decimorgan. This decimorgan. So Morgan is always capital M. So decimorgan, right? So this you have to remember the three important points. One is 1%, 100% and 10%. Is this clear? Okay, now the another one is a recombination. Now what is recombinations? We just now saw about the crossing over, because crossing over lead to the recombination because there is a new combinations is there, right? So here you can see if there is a no crossing over, suppose this is 1A and this is the B. So in these two conditions you can see if there is a no crossing over between the chromosome, this is a pair of chromosome, and if you can see crossing over cannot take place in this, so result, what result will be formed? Four non-recombinant chromatids are formed, right? And if in the second B diagram you can see if there is no crossing over but there is a chiasma formation take place, that means of crossing over between, actually it should be not crossing over, it's crossing over. So if there is a crossing over take place, there is a chiasma. With the help of this, what they are going to change, they are going to change their chromatids. So crossing over between these two chromatids lead to the exchange of their gene. So so when there is a combination between these two, so this is called non-recombinant, this is non-recombinant, but these two are called recombinant. So it's a recombinant chromatid, this is the non-recombinant chromatids. So there is two recombinant and two non-recombinant chromatids are formed, which is 50%, 50% combinant and 50% non-recombinant. So I can say 1:1 ratio, right? So crossing over result in the formation of new combinations of the character in an organisms, and that is called the recombinants. So in this segment of DNA are broken and recombined to produce a new combination of alleles. That process is called recombination. So here you can see how this recombination take place. If the crossing over take place, recombination will happen. So the combination recombinations, how you will calculate the recombination? So number of recombinant offsprings, total number of offspring into multiply by 100, you will get the recombination frequency. How to calculate the recombination frequency? So the number of recombinant is equal to, or I can write here for you, I think so, this will be help you, recombination frequency is equal to number of recombinant upon total number of offspring multiply by 100, as we studied in the previous, right? This is what the simple formula. Recombination frequency is equal to number of recombinant upon total number of progeny or the offspring multiply by 100. You will get the recombination frequency, right? And this recombination frequency can be measured in map unit or in the centimorgan. If there is 10, or I mean to say if there is a 1, so we can say the centimorgan and all, so that you can write 1 2 3 4 5 centimorgan, like that, so you can write it. Yes, so this is all about the today's session, what we studied about, that is the chromosomal inheritance of what? Chromosomal theory of inheritance, right? Along with that, we have also studied about the sex linkage and the recombinations and linkage mappings. So this is all about the today's sessions. If you have still any doubt, then please ask me. Otherwise, we will move on the question answer session. We will try to solve few questions based on what we studied today. Yes, anyone have a doubt, then please ask me. Otherwise, can I move with the next part of this session? Responded fast. I'm waiting to respond from everyone, not from only Anne. Okay, Bhuvanasri responded.
Sir,
Rahul responded. Great.
sir,
Yes.
this crossing over and recombination happens only when the traits are sex related.
Okay.
So, if other traits that are not sex related, they're always in complete linkage, sir.
See, always keep in mind crossing over take place in sex chromosomes, right?
Yes,
Yes.
sir. In other autosomes, there is no crossing
Hmm.
over, then it's called complete linkage. Since no crossing
Yes,
over takes
yes, we can
place.
see complete linkage because crossing over.
So all autosomes are complete linkage and allosomes might be incomplete linkage.
Yes, there is, we can say. Clear? Any other question? Okay, now the next part. Okay, now who proposed the chromosomal theory of inheritance? Sutton, Mendel, Boveri, and Morgan. Morgan and Mendel, Sutton and Boveri. Okay, so and Bhuana, then Kevin responded. What about Kirtini? Please respond it fast. Who proposed the chromosomal theory of inheritance? This is very simple question. Yes, it's a Sutton and Boveri. Next, the recombination of frequency between the purple and vestigial gene in the case of cross between a double heterozygous fly and homozygous recessive fly will be. Now here you have to find out the percentage of frequency. Now the percentage of frequency in between what? The double cross. So how you will calculate? See, simply for that we need to move on the diagram, like on this diagram you can see here, this is what the double heterozygous, this is a double heterozygous conditions. In that condition you can see if it is cross with the homozygous recessive for both the gene, what you will get? You will get a parental combination which is 1339. That means of diagram is also given there in that cross particular questions diagram will also given to you. So on the basis of that you have to calculate the percentage frequency. So it's very simple for you. The recombinant percentage you have to just total the recombinant frequency, right? So total when we add these two recombinant, how much it become? 5 4 plus 1 5, then 5 plus 5 10, right? So it become 0 and 3 not 5. Yes, I'm right. Yes, and you have to add these two parental combination as well as, so parental combination when we add, how much it is? 9 plus 5 it become 14, 9 plus 3 plus 1, so it become 13, so 3 reminder 1, then 1 plus 3 become 4, 1 5 and 2. Am I right? So this is parental combination, this is recombinant. When you add both, you will get, okay, so and told me the total 2839, right? 2839. So now you know the formula frequency of recombinant. So recombinant is how much? It is 305, and total number of offspring we just calculated, which is 2839 multiplied by 100. So how much you will get? Just multiply and divide, you will get 10.7%. So 10.7% means from the above picture you will get 10.7, means A is the accurate answer. Here this is how you have to calculate the percentage of recombination frequency. Are you getting my point? See, this diagram is given to you, this chart is given to you, and with the help of this chart you have to just calculate the percentage of frequency. Okay. Next question, which of the following is true about the linkage? It is the phenomenon in which more recombinant are produced in F2 generation. Linkage we have to talk about. More parental combinations are produced in the F2 generation. Genotypes which are present in F1 hybrid reappear in the high frequency in the F2 generations. It is the phenomenon in which two chromosomes are linked. So we have to choose the right option from this A, B, C and D. Now tell me which one is the correct statement, or we can say this particular question. Okay, so more parental combinations are produced in the F2 generation, but actually this is not a true statement. See, it is the phenomenon in which more combinants are produced in the F2 generation, right? More combinants are produced in the F2 generations. Why? Because rest of all three statements are incorrect. Why? Because less parental combinations are produced in the F2 generation. Here is talking about the parental combination. Is you will get the parental combination in the linkage? No, that's why this is the wrong statement. More parental combination. Parental combination means
Sir,
what? Actually it is talking about the recombinations, right?
sir, in linkage it will not allow crossing over, so how is more recombinant produced in F2 generation?
Why? See, it is more recombinant produced. See, in the previous diagram here you can see the more recombination means the number of recombination is higher, na, you will get, right? It is always higher, but it here in case of parent there is no any recombinations, but the option given as the more parental combination. No, parental combination is not. Parental combination is exactly copy of their F1 gametes, or we can say the F1 parents in the F2 generation, which is absolutely wrong, right? And even genotype which is present here in F1 hybrid reappeared in the low frequency of F2 generation, but here it is mentioned as a high frequency. No, it is always in the low frequency, and that's why this is also a wrong statement. It is a phenomenon in which two chromosomes are linked. So it is a phenomenon in which two genes are physically linked, right? Two genes are physically linked. It is not a two chromosomes are only linked. They are physically linked. So they are the linkage. So this three statement is wrong statement. Only first statement is, we can say, the right statement, because it is a phenomenon in which more recombinant are produced in the F2 generation. Clear? Yes, it's A. Okay. Identify the right fact about the strength of linkage. Yes, strength of linkage. Identify the right statement or fact about it. Strength of linkage. It is inversely proportional to the distance between the linked gene, and it is inversely proportional to the crossing over. Yes. So these two are, we can say, absolutely right. Both are the strength of the linkage. So we can say here both A and B. So C is the right answer. Okay. Now here, gene on the chromosome are physically linked first, and no linkage group in the organism is equal to their haploid number of chromosomes. Now you have to tell me that which statement is, which option is correct for this particular question. Yes, question number, this is self practice question, assertion and reasoning type of questions is there. So see, genes on the chromosomes are physically linked. I think so this R statement is true, right? Second, number of linkage group in an organism is equal to their haploid number of chromosomes. This statement is also true. Means both statements are true, but A is not the right explanation for the B. So can we say that A and R, actually A and R are both are true, but R is not the correct explanation of A. So we can say B is the right in case of this, because both are totally different, because it related to the linked gene and it is related to the number of chromosomes in the haploid organisms. Yes, so this is what the summary what we studied about the chromosomal theory of inheritance, linkage, that how linkage take place. Yes, so this is all about the today's session.